Professor of Ophthalmology, Biochemistry, and Molecular Medicine at the University of Montreal in Canada. Dr. Sapieha is the Canada Research Chair in Retinal Cell Biology and holder of the Wolf Professorship in Translational Vision Research at the University of Montreal, and the Chief Scientist at Unity Biotechnology. Dr. Sapieha is also the founder and Chief Scientific Officer of Semathera Inc. Dr. Sapieha received a bachelor's degree with a major in biochemistry and minor in management from the McGill University. Dr. Sapieha then obtained his master's and PhD both in the University of Montreal, focusing on pathology and cellular biology, being mentored by Dr. Adriana Di Polo at the University of Montreal. He then pursued postdoctoral training, being a fellow at the McGill University in pharmacology and therapeutics, and a second postdoctoral fellowship at the Harvard Medical School in ophthalmology. Dr. Sapieha has a longstanding research activity in the field, focusing on understanding the pathophysiology of proliferative retinopathies and age-related macular degeneration. His work has demonstrated the influence of innate immune memory and age-related neuroinflammation and the discovery of retinal ganglion cell participation in vascular pathogenesis and proliferative retinopathies. Dr. Sapieha has been a leader in the field, being the director of the Neurovascular Eye Disease Research Unit at the Hospital Maisonneuve, Rosemont. Dr. Sapieha has published over 100 peer-reviewed papers in prestigious journals such as Nature Medicine, Science, PNAS, and Journal of Clinical Investigation. Dr. Sapieha has also developed over 100 registered patents, 6 licenses to industry, and has founded biotechnology enterprises stemming from the research performed at his lab. He is dedicated to training the next generation of scientists, having currently trained 16 postdocs, 11 PhD, and 6 master students, 16 undergrads, and 18 highly qualified personnel since 2010. He has won numerous awards and distinctions for his work, including being a fellow of the Canadian Academy of Health Sciences and a member of the College of the Royal Society of Canada, the Outcome Research Institute Senior Investigation Award, and the Bifocus Innovation AMD Award, just to name a few. Without further ado, it is our pleasure to have Dr. Sapieha present his talk for today, titled Senolytics in Retinal Vascular Disease.
Thank you very much, Carolyn, and thank you for that elaborate introduction. Thank you so much, Grajan and Chris, for the invitation to come and present our work today. Yeah, just right there. Yeah. Okay, great. So today I'm going to talk about or focus really on one story that's been developing over the last decade in our lab, and also I think what I'd like to highlight is really this interaction between industry and academia, and how it's kind of critical to take ideas from academia, from the bench, and bring them to products. So I think ultimately, the way that I see this is, in your academic career, it's like the artistic part. So you're like the architect that draws like a weird looking building, and it might not ever be able to get built because it's not feasible. So then you need the industry side, which is very process oriented and optimized and good at finding critical paths. And so I think this interaction between the two is quite critical. So just before I go forward, I'd like to just acknowledge that the work I'll present today, and just stuff we do every day, is really a conjecture of having a lot of highly talented people that really span the gamut of skills. So going from highly knowledgeable molecular biologists all the way to people that are more in a marketing position. Now, I will disclaim these two conflicts of interests. I'll primarily focus on the work that we've been doing with Unity Biotechnology, but I will touch a bit upon some foreign work. So last year, I had the pleasure of backpacking with my 2 kids across Poland, and we stopped in this castle in Krakow. So Krakow used to be the capital of Poland until 1569 or somewhere around there. And in this castle, there was this huge tapestry that you see on the left-hand side here. And there was these 2 men talking to each other. And at the top, it was written Genesis 6. So not remembering what Genesis 6 was, I Googled it, and here's what it said. It essentially said that God said that we will only live 120 years. And I thought that was kind of interesting because at the same time, I was reading all these studies and working on projects related to longevity. And when you look at the actual hardcore data, God was exactly right. So there's essentially never been anyone since we've been recording that has broken this 120-year ceiling. But at every single time throughout our recorded data sets, we've always had someone that's been around 120 years old. Now, what has happened is that we've gained around 2 years of lifespan every decade. So you can see this on the graph on the left-hand side with the two big dips during the world wars. But probably more sobering is if you remove the top 8 most contagious or mortality from the top 8 most contagious diseases, we've actually done very little to impact mortality rates. And this is also kind of interesting because it means that we're able to develop drugs to increase the overall average age, but we're also living with a lot of consequences on our immune system of infections, viruses, and so on. And these certainly have a lot of impact in CNS diseases, and specifically in reprogramming the innate immune system. So my lab really focuses on 3 different areas. So one of them is this epigenetics, and this is where I'd say right now 80% of our group is really trying to figure out what happens to the immune system as we age, and how do early stressors in your life impact the way that your microglial cells, for example, react when you age. So microglial cells can live in your CNS for at least 20, 30 years. So we have this preconceived notion that innate immunity is very short-lived, but these are actually very long-lived cells. The other, and I'll talk about, is the development of therapeutics for vascular permeability. And what I'll really focus on is the cellular senescence concept. So in the aging field, like in oncology and immunology, we like to put things in a framework. So these are called the hallmarks of aging. And it's a way to really kind of break down processes that are occurring in a given situation. So in this case, aging, and again, immunology, oncology, they all have their own frameworks. And so we're trying to understand how this cellular senescence really impacts chronic inflammation and how cellular senescence prevents optimal tissue function. We work on the healthy retina. So I am in an eye center here, so I don't need to preach about the fact the eye is the most important organ in the body. But we do want to understand how the retina, like the one you saw before with the beautiful vascular architecture, becomes a retina like this in advanced cases of diabetic retinopathy. So as you know, diabetes is growing in pandemic proportions. Depending on what your stage of disease is, or depending on how many years you've had the disease for, you have at least a 60% chance of developing a form of retinopathy. So not necessarily this full-grown retinopathy you see here, but the incidence of diabetic retinopathy in patients affected with diabetes is tremendous. There have been a number of breakthroughs and dare say revolutions in the treatment of not only diabetic retinopathy, but first actually age-related macular degeneration. So the ones that are now close to a $14 billion market, our annual market, are anti-VEGFs. So these are drugs that really changed the paradigm. And also what they showed the industry is that ophthalmology, who before 26 was not really a very interesting field for drug development, because everything that was on the market was either repurposed steroids or so on. So there was not much of a margin made. And all of a sudden these drugs came on and showed that, wow, ophthalmology is actually a really big field in which not only can you impact patient health, but so you can also make a healthy profit. Now, the problem with the anti-VEGFs, as great as they are, is that in patients with diabetic macular edema or diabetes, depending on what stats you believe in, up to 40% of the patients don't respond optimally. And there's also this fear that vascular endothelial growth factor being a very potent neurotrophic factor, that if you sequester too much of it, you can actually cause photoreceptor degeneration. So this has been seen in the CAT trial in age-related macular degeneration. I'm not aware of any such study in diabetic retinopathy, but certainly these patients that are getting treated that have diabetic retinopathy are a lot younger, and they'll receive these treatments for a much more protracted period of time. So what else can be done? So I'll just, in the next couple of slides, give a bit of a framework of how people think about it and how we think about it. So this is a very classic pathway of drug discovery and development. So I took it from actually a malaria report, so from 2003. And the reason why I like it is because it really highlights the importance of academia, especially in the exploratory early discovery, all the way up to the preclinical transition. So this is where all your science gets translated, essentially. And this is also where the highest failure rates are. So it makes a lot of sense that, you know, we do this in our academic labs before we embark into this transition here, which is a lot longer and of course, a lot more costly. Now, the failure rates, again, are for malaria. So in ophthalmology, they're actually much higher in these early stages. And a way that this could look like, so ideally, it starts with an idea. So we're all big proponents of omics, but I think coming in with an actual theory and understanding a mechanism is important. Then you do the early exploratory discovery work. And then you, of course, have to de-risk the biology. And the best way of de-risking it is really to going through repositories. And this is where, you know, clinicians and scientists should be working hand in hand. And then you embark on the hardcore drug development platforms that, you know, often can take several years. And then eventually you go through the preclinical toxicology and then to the clinical trials. So this whole process could take around 15 years. If you're lucky, maybe a bit less, sometimes more. I'll give you an example of the first time that we did this. So in 2009, had this idea that in patients with advanced diabetic retinopathy, they have ischemic zones. So essentially zones where blood vessels have failed to regenerate into. And what you see right next to these zones is that there's pre-retinal angiogenesis, meaning the blood vessels are growing, but they're just growing in the wrong area. So why is that? So we spent a number of years trying to figure this out and came up with this theory backed up by mechanisms that retinal ganglion neurons, so the ones at the surface of the retina, are producing repulsive factors that are preventing these blood vessels from coming in. We identified one of them as being semaphorin 3A. Then we later found out that it was actually not only preventing blood vessels from coming in, but it was also causing a lot of vascular permeability, and especially in the early stages of diabetic retinopathy. So being a biochemist, I was able to draw up a couple of different, a library of around 20, 30 traps, or at this point, molecules that sequester or modulate activity of semaphorin and VEGF. And we were able to start a company called Sapieha. We brought this into efficacy trials. We were seeing that these retinas were revascularizing. This is all preclinical, that were revascularizing ischemic retinas and alleviating the pathological angiogenesis. So this thing here that essentially eventually becomes fibrotic and exacerbates disease. So, you know, taking this into clinic was the next step. Because it's a biologic, it's quite, it takes a lot of resources to produce versus a small molecule. So we partnered this program with, or we partnered the company with Roche, and now Sapieha is part of essentially the Roche pipeline, and it's continued to be developed by Roche in Switzerland and in Germany. So the rest of my talk will focus on another idea. So not to necessarily revascularize the ischemic retina, but try to understand how, you know, what exactly distinguishes pathological blood vessels, the ones that are leaky, that are growing in areas where typically you would not have any blood vessels, what distinguishes these ball-like structures from these healthy ones? And essentially, can we find a way to tag them and to selectively remove them? And could this actually aid in regenerating functional blood vessels? So can we have a disease modifying approach by eliminating the source of the disease? And so this is inspired partially by what happens to infants that have a disease called retinopathy of prematurity. So essentially in the US, for around 4 million births, around 15,000 infants have retinopathy of prematurity, but only around 1,500 of them actually develop a form of retinopathy of prematurity that is severe enough to require medical treatment, and only 500 around develop blindness. So this means that there's a very high remodeling capacity in these infants that have this preretinal angiogenesis. And essentially, the way that we can study these remodeling events in the lab is by using different animal models. And the one that we're using is the oxygen-induced retinopathy model developed by my former mentor, Lewis Smith. And so essentially, this is loosely based on retinopathy of prematurity. So we take mouse pups, put them in hyperoxygen for 5 days. They come out, they have these big avascular zones, much like you would have in a patient with advanced diabetic retinopathy. And eventually, because they're put back into room air, you start to see pre-retinal angiogenesis, much like you would see in retinopathy of prematurity, but also in some later stages of proliferative diabetic retinopathy. And now the part that's studied quite a bit is this part of how do these blood vessels grow. What's known, or what's studied a bit less, is how do they actually regress in this model. So this is a juvenile model, and these mice have a tremendous capacity to fix their retinas. And so can we learn something from these infant mice and develop therapeutics from this? So again, this model has a couple of different salient features of retinopathy. Firstly, that they have avascular zones, and second of all, that they have this pathological angiogenesis. So these are two readouts that we're always interested in. So when we started to try to understand what processes were at play during this remodeling, we found two things. One is that the neurons, so the ganglion cells and essentially the neural retina, go into a type of hibernation state. So they essentially stop producing most proteins for export, and they kind of focus on critical survival pathways. So producing proteins for export, loading ribosomes is one of the most energy intensive processes in a cell. So when these cells become hypoxic, no longer have enough metabolic supply, they really slow down production through a process called endoplasmic reticular stress, where they start to cut up to 300 or more different transcripts for export. Now, if this ER stress is not resolved, then the cells, including the endothelial cells, will trigger a pathway called cellular senescence. And I'll talk about that a bit later. But essentially, the cellular senescence, what we found in these studies, was that if you interfere with it, you can actually modulate the vascular outcome, and these retinas start to regenerate functional blood vessels. So this is what a three-dimensional cross-section of a retina looks like, or a flat mount looks like. And if you stain it for markers of cellular senescence in blue, you see that there's a high enrichment of these senescent cells that are present in the pathological angiogenesis. So not all of the cells in these tufts are senescent, but there's a good number of them that do express markers of cellular senescence. So what is this cellular senescence? So this is a concept that was coined in the early 1960s. And essentially it was Leonard Hayflick who was culturing fibroblasts. And what he noticed was that inadvertently, after 50 divisions, his fibroblasts would stop dividing. So essentially, he thought this was quite frustrating because he couldn't continue on with his experiments. But upon discussions, like, this could be interesting, and he published the paper as is. And we now know that this suggests that there's a program in cellular division or cellular longevity that can impact the vigor of the cell or the function of the cell. And we also know that what he was noticing is due to telomere attrition. And so this idea was kind of co-opted into the aging field, and cellular senescence became a synonym of aging, which is not fully wrong, but it's also not fully true. So we know that beyond just aging or cellular division, a lot of other stressors that you might encounter in a disease state, like hyperglycemia, heightened inflammation or persistent inflammation, irradiation, or unchecked proliferation. So for example, if you have a cell that divides too many times, eventually it'll trigger pathways of cellular senescence to prevent oncogenesis. So it's a pathway that's thought to have evolved with apoptosis to limit tumor growth. And another thing that happens to these cells is that because they have this high level DNA damage, they start to produce secretomes. So very staged secretomes, initially reparative secretomes. So they'll start producing a lot of IL-6 to help reprogram neighboring cells, fix them. And eventually they start to call for immune cells to come and clear them out and repair the tissue. Now, when this process fails, then you're in a state of chronic inflammation, and that's where the problems occur. And also, these cells no longer communicate appropriately with their neighboring cells. So we wanted to know what happened with these senescent blood vessels in time. And so we did some single cell RNA-seq data in that model. And what we found was that of the cells that we were looking at the transcripts, the ones that had the highest level of gene families coding for process of cellular senescence were essentially the non-neuronal cells. So the ones that can get recycled pretty easily, like pericytes, endothelial cells, astrocytes, to a certain extent, Mueller cells. And what was perhaps more interesting was that those cells that can get recycled were actually the ones that were producing all the inflammatory factors. And the neurons were actually not trying to produce inflammatory factors, probably because they don't want to attract microglia or other immune cells to remove them. Whereas these cells are a lot less expensive, so you can eliminate them and then the retina will regenerate itself. So we tried to find out what effectors of innate immunity were coming in. And to make a long story short, we found that neutrophils were actually coming in quite late during this process and throwing out their DNA content onto the endothelium that was senescent. So they're getting called in by senescent endothelial cells, and they're essentially throwing out their DNA, which is highly toxic to a cell. Extracellular histones is a major damage response signal. And eventually this kills or selectively kills those senescent endothelial cells. And we're able to build this story here where you have these pre-retinal neovascular tufts that have a good number of senescent cells, and you have the innate immune system that tries to clear them out. And essentially in these younger animals, the innate immune system is very effective. And could we find a way to mobilize this or to use this concept to regenerate retinas in patients with neovascular eye disease? And so knowing that we can't heighten the amount of neutrophils or increase inflammation in the retina, we thought we could perhaps do this using a small molecule approach. And so this work was inspired, and then essentially everything else I'll show you today was done with Unity Biotechnology up in the Bay Area. And it's inspired by work from Jan van Dorsten from the Mayo Clinic. And so these two mice are litter mates. They're genetically modified to harbor a suicide gene next to driven by the P16 promoter. So essentially P16 is the most fateful marker of senescent cells. And if you add a active compound to the mouse, you can selectively eliminate their senescent cells, or you can give them a placebo. And so these mice are 24 months old, which is roughly 70 to 75 years old in a human, if you can make that kind of comparison. And the mouse here received the placebo. and its litter mate received the active compound, which will selectively kill senescent cells by this apoptotic process. And essentially what you see is that the mouse on the left-hand side has several salient features of aging, like cataracts, osteoporosis. Its fur is nice. And also it lives around 18% less than the mouse on this side. So because that was a genetic experiment, we needed to take this and develop a small molecule approach. But first we wanted to see if the same things were happening in the retina. So we took those mice, ran them through our oxygen-induced retinopathy model. And what we noticed was that when we eliminated those senescent cells, two things happened. One was we were thinking expected. So because we knew that the pathological angiogenesis that you see in blue here is essentially a lot of senescent cells, by removing them, we were able to alleviate a lot of the neovascularization. So that's number one feature of the model. And the second thing that happened that we were quite excited about, but didn't necessarily expect, was that by removing those senescent endothelial cells, we're able to regenerate the ischemic zones. So we're essentially allowing, so it's like the senescent cells were a break to vascular remodeling or beneficial vascular remodeling in the retina. So to take this to the clinic, we needed to develop a small molecule. And the general concept is that if we can target senescent cells within a tissue, then those senescent cells will eventually die out, and the healthy cells next door will regenerate and reform functional junctions, and ideally the tissue will be healthy or rejuvenated, if I dare say. And then in the end, we'll have a functional tissue. So we spent a number of years trying to identify susceptibility nodes in senescent cells. And the ones that we were specifically interested in were endothelial cells. So the vascular cells lining the inside of your blood vessels. Oh, and essentially with all this work, we found that the BCL2 family, so B-cell lymphoma 2 family member, BCL-XL, was the most selective molecule or most selective effector of cell death in senescent cells. Did I just lose the Zoom? Okay. Sorry about that. I'll click them right here. Using passcode. That's it. Okay, so okay. Okay. Apologies. All right, so just to take that over, so we essentially identified that BCL-XL was the most selective node in senescent endothelial cells for survival. So non-senescent endothelial cells and non-senescent cells have redundant mechanisms built in, and when a cell becomes senescent, it loses these other survival mechanisms and exclusively relies on what we found to be BCL-XL. So we developed a family of small molecule inhibitors for BCL-XL and then characterized them. So here you see target engagement and mechanism engagement experiments. So essentially we know that we're engaging the target, so BCL-XL. And as we engage the target, we're activating apoptotic pathways. So essentially we're able to kill off these senescent endothelial cells. So this is a log scale. So we have around 1,000-fold therapeutic window between killing healthy cells and senescent cells. So essentially when we're dosing our animals and later our patients, we're really in a very safe therapeutic window. Now, when we treated the mice that were undergoing this active neovascularization, and profiled the retinas, what we found was that we're really selectively removing one single type of cell, or the one cell that was absent with all this single cell RNA-seq data, were these endothelial cells that you can see in this little cluster here that were expressing serpin and other markers of cellular senescence. So it gave us a lot of confidence that we had a selective remover or eliminator of senescent endothelial cells. So can we use this now to bring it to clinic? So the first disease that we wanted to go into was diabetic macular edema. And I'll show you in the next slides the reasons why we chose diabetic macular edema. So first of all, it's a highly prevalent complication of diabetic retinopathy. So at any stage of your diabetic retinopathy, you can have diabetic macular edema. And it's thought that the prevalence in the US is at least 1 million individuals. Now, when we looked at cadaver eyes from patients that had been diagnosed with diabetic macular edema, we found that the P16, so again, the most faithful marker of cellular senescence, was found in the superficial vascular layers, so associated with these endothelial cells at the top of the retina. And to a certain extent, we also found a fair amount of it in the retinal pigment epithelium. Now, when we looked at the vitreous, so I had the good fortune of having a postdoc in the lab who was a vitreoretinal surgeon, and essentially he would give us, every time he had a patient with diabetic macular edema or another vascular retinal disease or non-vascular retinal diseases as controls, he would be able to provide us fresh vitreous from right above the lesion sites. And we would profile this. And so some factors like interleukin 6, interleukin 8 that are not just associated with cellular senescence, but others like plasminogen activator PI1 were really highly upregulated in these patients. So this by itself doesn't tell you that there's a senescence process, but it doesn't disprove it either. It actually builds the senescence disease hypothesis in the diabetic retina. Now, then we went to models of mouse models of diabetic retinopathy. So the first one was the streptozotocin-induced model where essentially the toxin ablates beta islet cells. These mice develop some features of diabetic eye disease, so their blood vessels end up leaking at one point, and they do have retinal inflammation. So, no, I think I'm losing people. So do we reconnect? Sorry for breaking up here. Yeah, I'm sure. Oh, it's taking a while. No. Yep. Yeah. Yeah, no, it's super interesting. And actually, if you give low doses of LPS, so an E. coli polysaccharide, you can actually reduce neovascularization. So I think you're mobilizing the innate immune system, you're hyperactivating it. In the C3 inhibitors or C5 inhibitor by Apellis, you have heightened portal neovascularization. So we know that neovascularization is coded by complement. So it works super well with that theory that the immune system needs to recognize these neovessels and essentially ends up pruning the pathological angiogenesis. That's interesting. Yeah, possibly. Yeah. Although, yeah, I don't know. It's a pretty low prevalence event, right? So, yeah, perhaps. Sorry about this. Yeah. Bronson? Okay. No. No. 306. Okay. Sure, no, sure. General. Okay, so maybe because of that little break, I'll just quickly go through this. Essentially, we did single cell RNA-seq on diabetic retinas and found that there was an increase of one population of endothelial cells that we called endothelial cell 2. And these were the cells that were upregulated, or that when you profile their transcripts, what they were the most upregulated in were these transcripts that coded for cellular senescence. Now, when we looked at what else they were doing, they were the ones that had the less amount of junctional or transcripts that coded for junctional proteins like cadherins and so on. So the other things that we found were that essentially everything that was associated with barrier function was reduced. And with that, we're able to build this model. So essentially, where you have a given amount of endothelial cells that become senescent in the blood vessel, and then to kind of understand what these endothelial cells were doing, we used a process of peptide labeling called SILAC, where we would use different forms of amino acids that were labeled. And with that, we were able to see that what differed the most between a healthy endothelial cell and a senescent endothelial cell. So essentially, what were the peptides or proteins that were being preferentially produced by endothelial cells? And we found that most of them were actually proteins that were coding for cellular, for the immune system. So either cytokines or other adhesion molecules for the immune system. And so with that, we could build this model that within a microvasculature of the retina, when we have a senescent cell, we have these little factories of cytokines. Now, another thing that we found was that these endothelial cells, consistent with the transcriptomic data, were producing V cadherins, so these zipper-like proteins that hold endothelial cells together, but at a much lower rate. And most of them were actually trapped inside of the cell. So you could really kind of distinguish this senescent endothelial cell. So this is an endothelial cell that essentially looks like if you take an egg and break it onto a frying pan. So the senescent cells change shapes. They have a flatter cytoplasm. And what we're finding were that these zipper-like proteins were actually integrated or not getting pushed to the cell surface. So when we looked at impedance measurements, so essentially we made monolayers with increasing amounts of senescent endothelial cells and then measured the capacity of those monolayers to hold ion flux. So the lower the impedance needed to cross the membrane, the more permeable these membranes were. And what we found was that the more senescent cells we had, the more compromised or the less barrier was functioning. So we built this model where you have a number of senescent cells within your vasculature, and they're essentially allowing vascular leakage. And so you can imagine that if you had only a couple of these cells within a microvessel, it could still cause vascular leakage. So then using the, again, the mouse model, we looked at two things. One was retinal inflammation. So you can see here that these diabetic mice have significantly higher levels of retinal inflammation. So interleukins 1, 6, and TNF alpha. And with treatment of our BCL-XL inhibitor, we're able to eliminate these senescent endothelial cells, and hence we're alleviating the microinflammation produced by these endothelial cells. And with this reduction in microinflammation, eventually the full retinal inflammatory status is improved. Now, another thing we looked at was the barrier function. So again, the capacity of those endothelial cells to hold fluid within the blood vessels. And you can see in this model that diabetic mice actually leak albumin at a much greater rate. So around twofold dynamic range. So what this means is that if this leakage continues, then eventually the retina swells. So mice don't have a macula, but we look at the amount of extravasation. And with our UBX1325, so our BCL-XL inhibitor, we're able to very significantly reduce this amount of leakage. So, and by reducing this leakage, what we think is happening is that we're eliminating senescent cells, and the healthy neighboring endothelial cells are regenerating and reforming proper junctions. So this was work that was done by a CRO and by us simultaneously. So it gave us confidence that both groups were always finding the same thing, and it made us go into clinical trials. So I'll save you the safety profile data. So essentially, there was almost no tox noticed in preclinical models. And when we looked at the treatment, the TAEs, there was almost, it was a very, very clean tox profile. So the patient population that we went for were patients that had previously been treated with anti-VEGFs, but were not responding optimally. And what happens with anti-VEGF treatment is that there's a plateau that's reached after around 6 months of a treatment course. So there's these initial gains in best corrected visual acuity, but eventually there's a plateau that's attained. And these patients actually never really get better after 6 months, or they rarely ever actually gain vision after, or so the data says. So just for those that are a bit less familiar with BCVA, so best corrected visual acuity is measured by these logMAR vision charts, and a 20/20 vision is a BCVA of 85. What you need to drive in most states is 20/40, so a BCVA of 70. And this is an example of charts of patients receiving anti-VEGF treatments. And so you can see that the treatment regimens call for injections anywhere between 4 weeks to 3 months now, depending on what the patient's on and how well they're responding. And so you can imagine that these patients are receiving a lot of injections in their daily lives. And what we took, the patient population we took into the clinical trial, we wanted them to have at least 2 injections in the last 6 months. The actual number was 4 injections in the prior 6 months. And these are patients also that had a high level of residual fluid, so around 440 microns. So the retinas and their foveas were swollen. When we took patients that had visual acuity of 61 letters. So these were patients that were not doing well. And what we did was we gave them 1 single injection. So again, these patients had received 4 injections the last 6 months. We gave them 1 injection and then followed them for a full year. And here's what happened. So at least 53% of patients did not require any other treatment after this 1 UBX injection. So again, these were patients that were receiving on average 4 injections in the last 6 months. And then with 1 injection of UBX 1325, they were able to go at least a year. So we stopped the trial at a year without receiving any other treatment as deemed by their ophthalmologist. When we look at the actual gains in vision, we had a delta of 7.6 letters. So that's essentially a line and a half on a chart. And patients with UBX were significantly better than their shams. Now, interestingly, when we looked at the sub-analysis, so we're not claiming that the worst patients are doing best, but this is just an example of your improvement versus baseline. So these were patients that had the lowest amounts of BCVA, so the worst, the ones that were seeing the worst, and they were gaining around 10 letters after a single injection. So a year later, this patient subgroup had 10 letters more. So if you need 70 letters to drive a car and you're like just around 60, you can imagine that with an injection a year later, you're still, in that range where you could be driving your car. So we were quite excited by that. When we looked at the central subfield thickness, so essentially looking at the amount of edema in the retinas, what we found was that the UBX patients were stabilized. So they didn't necessarily drop in their CST, but they didn't seem to gain any more edema. So we think this is interesting, and we're trying to understand exactly how they're not gaining, but they're not getting worse. So we're stabilizing patients, again, with 1 injection a year in. This is a patient that responded well. And so again, this patient had received 4 doses in the last 6 months. And so clearly not responding to anti-VEGF treatment. So 4 doses of anti-VEGFs before. And then with 1 injection of UBX 1325, you can see that their subfoveal thickness is actually getting restored to what you would see in a healthier individual. And eventually, after 6 months, this patient is starting to get some cystoid macular fluid. And but certainly in these early phases, they're doing quite well, and they're also gaining a significant amount of letters, so up to 10 letters, this patient. So we're now fully enrolled our phase 2b trial. So we're going head to head with the aflibercept, and with our data coming out in sometime in the first part of 2025. So this is a trial that has 52 patients in it, and we're trying 3 doses, so staggered every 8 weeks. So we know that 1 dose works very well, and we'll see if 2 doses can perhaps afford a better efficacy, or 3 even better. And we'll know that shortly. So with this, we can build the following model where we have endothelial cells that become senescent. So subsets of endothelial cells in the diabetic retina that become senescent. With UBX 1325, we're able to eliminate these cells. So things like microinflammation, or the fact that they're not able to form proper junctions with each other, can get repaired if the neighboring endothelial cells replenish these cells. And ultimately, we think we're getting such long durability because we're repairing the retinas. We have a disease modifying approach. And then just to finish off, so I think with the technical issues a bit over, I'll show you a little video that we had made to kind of explain what we think is happening. So essentially, as a summary, what we think is happening is that these in diabetic eye disease, we're getting subsets of endothelial cells within the blood vessels, so around the macula and also peripherally, that are triggering pathways of cellular senescence, and they're essentially not able to form proper junctions with their neighbors. So you get edema, and also they're producing cytokines that cause microinflammation. So we developed UBX 1325, which binds and inhibits proper function of BCL-XL. And this essentially sends the senescent endothelial cells into an apoptotic cell death. And when those cells get eliminated, the healthy cells next door can regenerate. And this leads to reduced retinal inflammation and reduced vascular permeability. So we're getting increased barrier function. And another thing that we're noticing in preclinical models is that the blood vessels are actually remodeling and regenerating into avascular zones. And collectively, this leads to better vision and better drops in subfoveal thickness. So with that, I'd like to thank everyone that participated in this work. So people from my lab in Montreal and people in at Unity Biotechnology. So all the clinical work was done with Unity, a couple of hours north of here. So I'll be happy to take any questions.
Mike, thank you very much. We will start from this side of the room.
Thank you. Really enjoyed the talk. My name is Jan. I'm Professor Brown postdoc. I wonder about your endpoint. Is there a specific reason why you chose visual acuity BCVA, given that you reach plateau? My question, just to give it a bit more meat, for those of you who don't know, to pass one line in an ETS chart, you need to get 5 out of 5 correct, meaning 10. So single letter count doesn't really make sense, given that 5 have the same dogma step. Is there perhaps any other data you could show on controls that you show controls don't show learning effects, but your treated patients did?
Sure. So the reason why we chose BCVA is for your FDA approvable endpoint, you need function. So that's perhaps changing now with approval of some of the GA drugs where structure could potentially be used. But right now, what we know is it's got to be a functional endpoint. So that's the reason why we chose BCVA. Within the noise, so I mean, I think what we're seeing now, there are certainly meaningful changes in the BCVA. The 5-letter gain versus what we see in the treatment is around 5.2 letters, and then we're seeing drops in the sham. So the delta is actually around 7 to 8 letters. Yeah, so I think that varies from site to site. Yeah. Yeah. Yeah, so we did other functional endpoints like MFERGs that were ITTs, so investigator-initiated trials. And we did that in another trial for neovascular AMD. And then we found, subsets of patients that had received the drugs. And this was a aflibercept, head-to-head aflibercept trial. So we did see improvements in MFERG. So again, MFERG, I don't think you can build that into your pivotal trials. other things we're thinking about is microperimetry. But again, the BCVA, I believe, is the most widely, I actually know is the most widely used endpoint for function in these trials. Mm-hmm. Absolutely. Yeah, absolutely. BCVA. Yeah.
Mike, very nice talk. So I have some question about senescent cells.
Mhm.
So if I remember your single cell, there's like 3 different state of endothelial cells.
Yeah.
And in the control, they also have some senescence
Yeah.
cells. I'm wondering, like, why only small portion goes through senescence? Are they localized, for example, at the tip of the vessel, or just what's the heterogeneity come from?
Yes, that's a great question. And we're looking into that because initially we thought there would be a much bigger shift. But we have around like a 2-fold shift in that one cell population that codes or that's enriched in transcripts for senescent cells. So I think if we kind of model it, we'll probably be getting around 8 to 10% of the cells within the blood vessels, that endothelial cells that have these pathways augmented. So it means that in a healthy blood vessel, you probably have, anywhere between half or less than half of that. So what happens to those other cells? Good question. Are they triggering? So it's transcriptomic data. So it means that those cells have not necessarily committed to a senescent pathway, but they're starting to regulate genes for that process. So could they get themselves just naturally turned over or eliminated? So that's something that we'd be interested in learning about.
From the audience. So first, Tim Kern.
Oh, yes. Hi, Tim.
Hey, so your therapeutic approach is remarkable, but my question is, what makes the retina uniquely susceptible to the lesions of diabetic retinopathy? Are these senescent cells, are this senescent process unique to the retina? Does that mean that are local factors involved?
No. So to answer your question, it's quite a ubiquitous mechanism, and actually adipocytes trigger pathways of cellular senescence at very, very high rates. And if you eliminate the adipocytes, you can actually alter your course of diabetes. So it's certainly not specific to the retina. One of the maybe to kind of expand on that question, the concept of systemic senolytics is something that's much of a hot topic. Now, the issue with the systemic delivery of these senolytics, for example, the BCL-XL, we know might have liabilities for platelets. So there have been clinical trials with navitoclax, which inhibits BCL-XL, but also BCL2. And those trials were discontinued because of neutropenia and low platelet numbers. So for a senolytic, we don't need an aggressive regimen as you would in oncology. So it's feasible eventually to have, a systemic regimen that can just eliminate the right amount of senescent cells, and then you go on, a drug holiday for 6 months and then get another treatment.
Thank you. And another question from Narcissa.
Thank you very much for this very interesting translational research. My question is, don't you think that establishing the functional BCVA to just 20/20 will make you miss some improvements in patients having better than 20/20 vision? Because we know vision can be much better than this inferior lowest limit of normal.
Mm-hmm.
And if you establish to 20/20, you did not record, or the nurse, whoever recorded the visual acuity, did not test the patient if they can see more. We have many cases with diabetic retinopathy and macular edema that come sometimes with vision 20/20, but they have significant macular edema. And with the injections, we can restore a possible much better vision than before the onset of macular edema.
So interesting concept that the patients that we were enrolling had BCVAs that were significantly lower than 20/20. So they were, their or the equivalent of 20/20. So their BCVAs were around 60, the actual. So if we brought them anywhere into the zone of 20/20, we would have had, a pretty good home run. I think certainly we didn't plateau the reads at 20/20. If it went above that, that would have been registered.
So you recorded better vision than 20/20, and definitely there was nothing to improve better than that. Because
we
the visual acuity has a normal distribution. So visions better than 20/20 in a large clinical trial or fairly large clinical trial will definitely be between the best patients' vision.
Well, yeah, so nobody in our trial.
Yeah.
All right, let's move on the next question.
Nobody in our trial hit 20/20. So they're so far below that they never made it to that level.
Thank you very much for presenting this amazing work. More philosophical question. So is the senescence a like a natural, like a healthy way of aging, and what goes wrong in your normal body? And when you use a senolytic agents, is it making more like a wound healing response, or the cells are? I'm just trying to understand the big picture there.
Yeah.
Thank you.
Yeah, so that's a great question. It's more than philosophical. I think there's actually some nice biology to answer that. So so it's thought to be part of initially a repair mechanism or an anti-proliferative mechanism, so to prevent oncogenesis, like I gave an example of. So with the healing process, I think this is probably what's happening mostly in aging. So you end up getting successive hits that eventually build up in your cell, then the cell becomes senescent, and it starts to produce a very staged like secretome. So it starts secreting reparative factors initially, and so there's some people that claim that at any given time, if you look in the liver or other tissues, you'll have 2 to 3% of your cells that are senescent, and they get turned over. So if you induce an injury in the liver, for example, you'll have some cells that are senescent. They're going to be reprogramming the neighboring cells, and then eventually natural killer cells can come in and eliminate them. No more senescent cell, and the tissue is healed. So this is a very beneficial way that cellular senescence functions. The problem is, as we age or as we have general immune compromise and diabetes or other systemic diseases, your immune system is no longer able to clear those senescent cells, and then they start to accrete in the tissue. They also upregulate like PD-L1, for example. So it's like they're actively trying to evade at one point immune surveillance. So I think this is kind of where senescence becomes bad. Is like inflammation. Like if you have acute inflammation, acute inflammatory stress, well, that's good, right? You're repairing the tissue, you're getting rid of your infection. And then if it's sustained, that's when things go bad. So it's the same thing with cellular senescence. So if you have cellular senescence, you reprogram the neighboring tissue, then and then your cell gets removed or replaced, then that's likely kind of the ontology of the process.
Oh, hi. My name is Hewitt Antena. My question is about the efficacy of BCL inhibitors. So what % of patients get better? And do you see heterogeneity between senescent populations between patients that do respond to BCL inhibitors and those that don't?
Yeah.
And if so, what's the prospect of using a combination of therapeutics in addition to BCL inhibitors, so other senescent associated factors?
Yeah, so those are two great questions. So to start off with the patients that respond the best, so we have sub-analysis in our patient groups. We know essentially as a general, the worse the patient is, the better they respond. However, there's a nuance in that. So when you look at their glycated heme, so the A1C, so the patients with the best controlled glycemia actually respond better. So that's like the one exception. In terms of their BCVA, so I showed an example of patients that had BCVA under 60 that were gaining 10 letters. When you look at the full sub-analysis, actually patients that are closer to 70 are responding the best. Patients that had the highest CST, so that had the most macular fluid, are responding the best. So we're still trying to figure out exactly what the ideal patient profile is. Now, whether we could use this as an adjunct therapy to an anti-VEGF, so absolutely. I think one of our questions we had is, would we compromise some of the vascular remodeling? So we know that patients that are receiving anti-VEGFs never actually see any kind of vascular regeneration, or again, there's I think the meta-analysis really suggests that they're just not regenerating any vasculature. So if they have avascularity, it's not improving, and it makes sense with the mode of action of anti-VEGFs. So one of our fears was, would we compromise that remodeling capacity with the BCL-XL? And our preclinical data suggests that you can put both of them together. Also, if you treat infants with retinopathy of prematurity with an anti-VEGF, they actually regenerate their blood vessels quite well. So there's some processes in younger populations that, and our preclinical data together, that suggests that you could go both together.
Hi, my name is Sonia. I'm a researcher at Dr. Pachowski's lab. I had a question on hypoxia-inducible factor 1 alpha and its relation to VEGF. Are there any recent insights on how, let's say, patients that are displaying retinopathy due to hypoxic conditions, that you could manipulate HIF-1A and not directly going to VEGF? Are there any significant?
Yeah, so I think most of the work that I'm familiar with in that field was done by Akrit Shodhi from the Wilmer Eye Institute. And so he's showing a big stabilization of HIF in diabetic retinopathy and post-mortem eyes and so on. And they developed a small molecule that targets HIF-1 alpha. And it hasn't gone to clinic yet, as far as I know, but certainly in the preclinical models, it looks favorable. So HIF-1 alpha will not just regulate VEGF, it's going to regulate, Craig-Shimensah says 3,000 genes in your genome, so like a tenth of your genome. But so I think the HIF-1 inhibitors will be quite interesting to see how they pan out in clinic. But certainly in preclinical models, they seem to be working.
Very inspiring talk. Thank you very much. I'm Xuan from Dr. Ray Chen's lab. I have a question that, do different cell types that undergo senescence have shared markers and common therapeutic targets, or they're actually different? And do you have you observed other cell types that were targeted by the small molecule? Thank you.
Yeah, so that's another great question. So in terms of the cell activity, so we're not able to kill epithelial cells. We're not able to kill a whole set of other non-endothelial cells that are senescent with the BCL-XL inhibitor. So we're not fully sure why that is. One of the theories is that there's a compensatory upregulation of like MCL1 or other survival factors. And so initially, we thought, oh, this is unfortunate. We're only killing one type of cell. And then, when you think about it, it's actually kind of a benefit, because then you don't necessarily want to kill every cell that's got a senescent signature in your body. So I think the future in senolytic development will be to identify what are the susceptibility nodes in different cell types, and whether, this stems from the lineage of the cell, if it's like epidermal, endodermal, or ectodermal, like, does that have anything to do with it? But certainly a senolytic is not a senolytic for all senescent cells. And in terms of their signatures, they also differ. There's convergent signatures, like P16 seems to be pretty convergent, but then they vary a lot in terms of other cell cycle regulators, like P21 or so on. So yeah.
Hello, my name is Rafał. I'm postdoc in Chris's lab.
Nice.
I think my question is partially answered, but looking at your wonderful results, I was wondering if your senolytic drug can be repurposed to other diseases which have senescence in the mechanism. Thank you.
Yeah, thank you for the question. So, well, the easiest place to look at for us is other vascular diseases. And so it's certainly something that we're thinking about. Again, because we seem to be pretty unique to endothelial cells, it would have to be diseases that are characterized by a pathological burden of senescent cells in the endothelium. So we have some ideas for that. And then we would need to probably go systemic for a lot of those. So, any kind of cardiovascular indications or so on. But certainly something we're thinking about, but also thinking about just broadening the senolytic library.
Thank you for that really interesting talk. So this is more so out of pure personal curiosity, but do you see in your models of diabetes-induced endothelial senescence, like localized changes in microglial activation or microglial cell state? And is that reflective of the proportion of senescent endothelial cells within that area of vasculature? And that kind of ties into if there's heterogeneity of endothelial cell senescence in the diabetic retina.
So that's a great question. I'd love to be able to answer that, but I don't have an answer. I would think so, because again, because they're so active in their secretome, you would think that they would at least recruit selectively microglia or reprogram them to a certain extent. But there's no data that I'm aware of on that. That's an excellent research question.
Do you eventually foresee kind of targeting immune signaling axes as well to potentially increase the efficacy of your senolytic drugs?
Yeah, so I think, the whole immune senescence concept or field, I think, is a bit clouded, so I think a lot of pathways or markers that you would see in a senescent cell that's clearly not healthy, like a senescent endothelial cell or senescent epithelial cell that's clearly not healthy, mononuclear phagocytes actually use them for regular function, so people will say hey, look, there's this P16 positive microglia cell, or it's senescent. No, it just needs that to function normally, so I think it's been a bit clouded. So I think eliminating immune cells with senolytics is an interesting idea. I don't think we know enough about the biology to do it. But on that note, venetoclax, so this is a BCL2 inhibitor, is actually a standard of care for AML. So we do know that there's toxicities in immune cells with BCL-XL inhibitors, but is that attributed to senescence? Not clear.
A great talk. So my understanding is the drug is targeting senescent cells for apoptosis, right?
Mm-hmm.
So what will happen if the drug is targeting another pathway other than apoptosis in the senescent cells? Like if they are still there, activated them somehow and did not get cleared out by the immune system.
So you mean if the drug is as advertised inhibiting BCL-XL, but the cell is not going into an apoptotic pathway?
Yes.
So like pretty much every cell that's not senescent, right? So yeah, good question. So we have looked at that, of course, and the cells survive. So if they're not senescent, they survive, and we're exploring. We have some data now on shifts in their activity, but I'm not at liberty to discuss that further. But yeah, but they're but certainly the drug, for example, in the eye stays for 33 hours, or that's the bio half-life. So within 5 days, it's eliminated. But the time that it's in there, we don't see any negative impact on non-senescent cells. But if you do these experiments in a culture dish with a high dose of the drug, then yeah, there's certain changes you'll observe.
Hi, I am Marco from Kaiser Lab. Can you make a cancer stronger with your drug?
Yeah. So you mean can you potentiate cancer growth by eliminating senescence? Yeah. So that's another really thought-provoking question. So cancer cells become senescent, and it's thought that if you have too many senescent cells within a tumor, because of the secretory phenotype, you're actually going to accentuate your tumor. So the data there is primarily from preclinical models again. So if you use navitoclax, so like the kind of the prototypical small molecule senolytic, you'll slow down solid tumor growth. Primarily primarily is not clear, but by eliminating senescent cells, but also by killing off some of the, depending on what kind of tumor you're looking at. If you're looking at a like melanoma cell lines or so on, you'll kill the actual primary tumor also. Now, whether senescence is always good for cancer, it's initially good, but then it's got to get removed. If you have too many senescent cells in your tumor, those tumors progress more rapidly.
Great, thank you so much. I really enjoyed that talk over here.
Oh yeah.
So in people who respond poorly to anti-VEGF therapy for diabetic macular edema, using steroid is kind of what one goes to. Is this kind of shotgun effect, changing transcriptomics entirely in kind of indiscrete way. My question is, do you know if there's any effect on the BCLX pathway from steroids, whether it be temporary or not?
Yeah, that's a great question. I would think that with like dexamethasone implants or so on, you're one of the stressors that causes cellular senescence, which is bathing in a soup of cytokines. So this is called secondary senescence. Would probably be suppressed. Now we also know that hyperglycemia, for example, is a very, very slow inducer of cellular senescence. So if you think of any other stressor that can send the cell into senescence state, like irradiation is like the fastest one, free radical stress, like through hydrogen peroxide, modeled through. So all those stressors take in a dish around 3 days for your cell to become senescent. Hyperglycemia takes at least 2 months, or a month and a half, and you have like 20% of your cells in your dish will become senescent. So it's very, very mild, and likely that's evolved to protect against like postprandial spikes in glucose. So our endothelial cells actually have a very high capacity to buffer against becoming senescent. So by the time the patients that we're treating, they've had diabetes for 17 years, and they've been diagnosed with diabetic eye disease for 4 years. So it's a very slow process before you actually induce senescence in them. So I think if you use the dexamethasone implant or an injection, you would probably slow the process down. But the question is the damage already done before you're there?
All right, we had many simple, easy questions. Now let's move on.
That's great.
To thank Mike for his wonderful presentation today.
Well, thank you very much.